Welcome back. Today we are going to take up another topic, that is the pre-formulation studies. Why do we need to do pre-formulation studies? Goes one step back, that before we go to the development of the dosage forms, the complete physico-chemical, biological, microbiological, immunological characterization of the drug needs to be done, and this has to be done in compatibility with the excipients which would be incorporated into the dosage form. So, pre-formulation analyzes the drug in combination with all the excipients which are going to be used, so as to come out with the best combinations, and based on the output of the studies, the dosage form which needs to be developed based on the properties of the physical, chemical, biological, microbiological or immunological property, the best dosage form that can be developed is decided, and then it is taken further for the clinical trials and commercial marketing. So, it would be the first of the lectures of starting towards the creating the basis for the dosage form development. So, we will look into the next few slides what all characterization studies we need to do. Okay. So, why do we need pre-formulation studies? What are its applications? What are the various characterization studies which are performed, and finally, what we learned from that. Now, as I said, the pre-formulation studies is something learning before doing. What do we understand learning before doing is, we need to understand the physical characteristics, chemical characteristics, like what is the solubility, what is the particle size, what is the surface area, what is its tendency to absorb moisture, what is its tendency to be destabilized in the presence of moisture or in a specific environmental condition. So, we need to, what happens when it is put with some other excipient or ingredient, a chemical ingredient, whether it is going to be stable, whether it is going to destabilize or it is going to form a adduct. So, the pre-formulation studies gives us an idea of all the characteristics. That is why these studies are very beautifully described as learning before doing. The set of studies required before we go for the launch of any dosage form or any drug. So, the drug, that is the active pharmaceutical ingredient, or the drug product, that is the dosage form like tablets, capsules, which are designed around the drug. So, the designing of the drug product and the drug, that is the molecule which is responsible for any pharmacological action, requires extensive studies to study their properties. So, until unless the properties are not known, we will not be able to get a suitable dosage form of it, or even if we get a dosage form, it is not going to be stable, and it might not be working good enough in the patient. So, without having a knowledge of the characteristics, properties of the drug, if we develop, it would be like going 2 steps forward and then 4 steps backward to do corrections in the dosage form, and then coming back to designing a new dosage form, then encountering some issues. So, instead of going that way, the pharmaceutical sciences have evolved in a way wherein the complete physico-chemical characterization of the drug is done, and then the dosage form development is taken forward, so as to have a stable as well as a bioavailable dosage form. Now, so to summarize, the objectives of pre-formulation studies are to establish the physical and chemical properties of the new drug entity, to determine its kinetics, how is it going to behave in the system, that is, whether it is going to degrade by first order kinetics or second order or pseudo first order, or it is going to be, how is it going to be destabilized. So, the stability studies, then with what all ingredients it is going to be compatible. So, a list of all those ingredients, obviously the ingredients which need to be used, supposedly used along with the dosage form. So, the compatibility needs to be studied. Then it also tells you about how it needs to be processed, that is whether we should go for suppose if the drug is thermosensitive, that is in the chemical characterization, in the physical characterization it came out that the drug is sensitive because of the drug, it is thermolabile. So, can we use high temperatures or thermal based techniques for its processing, so that idea evolves when we go for these characterization studies. Also, if it's thermosensitive, what is the proper storage condition which should be proposed for the drug, so that the ingredients as well as the drug is stable and they are bioavailable and they do not cause any side effects in the patients who are taking. Then it is to estimate problem arising during formulation. That is, suppose there is some issue with the solubilization, or if even it's working properly in vitro, whether when when we take it into the in vivo clinical trial phase and it is given to the patients, so they might not be giving the same performance in as compared to the in vitro studies. So the pre-formulation studies are done again so as to find out the right particle characteristics, so that or the ingredients which need to be incorporated, or any solubilizer which needs to be incorporated, so that it's in vivo as well as in vitro comply. That is, there is a coherence or correlation between the in vitro dissolution as well as in vivo solubilization. Otherwise, it would lead to poor bioavailability. And the last but not the least, it is the entire basis of these studies is to develop a suitable dosage form which is going to be stable, bioavailable, and it is not going to cause any side effects, in terms of that the probability of sending the drug to the target area is max. Now, as I said, the complete phase in which starting from the pre-formulation till the marketing, there is a complete rigorous list of studies which need to be done. So once the drug or the active moiety is got, the preclinical studies are done, that is along with the pre-formulation studies to determine the best formulation which can be used. Now, it is not the finalized formulation, it will go on improvising. So based on the pre-formulation data, the best formulation is designed, it is studied in the preclinical studies, then the if the results are positive, they are taken positive in the sense that there are no side effects, absorption is good, permeability is good, distribution is good. So in totality, the bioavailability of the drug is good, which is calculated based on the pharmacokinetic parameters of the drug. And then it is taken to clinical trials, that is volunteer studies. Now, herein, since it is being extrapolated from the animals to the humans, there might be a need to change a few parameters in the formulation so as to suit the biological setup of the humans. So the pre-formulation studies are fine-tuned again so as to suit the human trials. Then in the phase 3 clinical trials and clinical studies of the trials, the best formulation is taken. We might be studying on 2 or 3 formulations or doses of formulation. Maybe one is parenteral, or one is a tablet, one is or tablets of 2 strengths are being studied. So the best formulation is taken up, and then these are sent for in the next phase, based on again positive bioavailability data, no side effects, no toxicity, the immediate toxicity. We are not looking into long term toxicity. It is sent for again the large scale manufacturing is done, and then it is launched. So this is how the pre-formulation is simultaneously being conducted at all the phases, because maybe that drug is getting absorbed in the animals, but it's not getting absorbed in the humans. So we need to add a few solubilizers, because the gastric juice contents, the pH conditions both vary in the animals as well as the in comparison to the humans. So fine tuning keeps on happening, and those all become a part of the pre-formulation studies. Now, so pre-formulation is an analysis of the macromolecular system prior to preparation of the formulation. Now, we need to identify the physical, chemical, mechanical, physicochemical and physico-mechanical, then how the drug might be absorbed. So there are in vitro permeability studies so as to help in the design of the appropriate dosage form. Suppose the permeability of the drug is very poor because of being highly hydrophilic. So, we need to design a formulation which might be injected into the body directly, or it may be converted into a dosage form which is, for example, if it's like a topical delivery, or so maybe that it can be converted into a ointment which is of hydrophobic base. So, depending upon the properties of the drug, during the pre-formulation state, the decision of what sort of a dosage form can be developed is taken. Now, the decision of what needs to be done is from both the objectives of how it is going to behave in vitro, that is outside the body while being on the shelf of the stores, and how it is going to behave in vitro. So, the pre-formulation gives us the basis for development of the drug product from both the aspects of in vitro and in vivo performance. Then, as we saw, it is also from the large scale processing, transportation, storage point of view. As the objectives we have seen, that is, it is to establish the physicochemical parameters of the new drug entity, with what all excipients it is compatible, what is its stability and kinetics. So, we will now look into parameters of what all kind of chemical moieties which are proposedly the drugs, mostly the new drug entities, how they are taken and how they need to be studied so that the most viable dosage form can be manufactured. So, the first in line is, suppose we have the drugs. One is the drug, that is the drug is the chemical entity which directly goes and produces the impact in the body. The other is the prodrug. The prodrug are synthetic derivatives of drug molecules that may have intrinsic pharmacological activity, but they need to go some sort of transformation in vivo to liberate the active drug. For example, erythromycin acetylate is esterified in the body so as to release the erythromycin. So, the reason is that it is not stable in the gastric juice, in the parent moiety, that is erythromycin. So, it is converted into acetylate form. Now, during the pre-formulation and the extent of how the two moieties can be separated can be studied. So, so as to improve the stability, solubility, whether if it is crystalline, so it might irritate the lining. So, the pharmaceutical improvements in terms of the physical and chemical properties of the drug can be done by during the pre-formulation stage. So, formulation of prodrugs is one of the techniques of doing these things. So, by formulating prodrugs of the drugs, we can create stable and new drug moieties, we can increase or decrease the solubility, we can play around with the crystallinity, we can, suppose some drug is very bitter, so we can convert it into sweeter moiety and modify its taste, color, odor, if it induces pain upon injection. So, all these things can be done when we are converting the drug into a prodrug form. Now, during high throughput screening, the new chemical entities are identified which are probably going to have the high probability of having drug like properties. Now, so these are shortlisted. So, the lead new chemical entities are selected and they are then further taken up for suitable dosage form optimization. Now, this is done in two phases. One is the therapeutic formulation development, the other is the toxicokinetic studies. So, what we are trying to understand here is, once the NCE is identified, it might not be suitable from the point of view of formulating it, or it might have some toxic properties. So, we need to go on optimizing the drug the individual functional characteristics, chemical properties of the drug, so that its toxicity is reduced. Suppose if it is highly lipophilic, we can add some moieties so that its water solubility is improved. So, the pre-formulation testing is simultaneously done at all these stages so as to improvise the physical, chemical and biological properties. Now, pre-formulation testing is done from the physical point of view, that is the size, shape, hygroscopicity, crystallinity, rheological properties, flowability. It is from the regulatory aspect, because we want to go for the filing of investigational new drug, then the abbreviated new drug application, chemical or the analytical testing point of view. If the drug is having chiral properties, so it might be active or it might be non-active. So the pre-formulation studies is done from all these points of view, so as to have a compound or a compound which is a new chemical entity, so that it can qualify to be characterized as a drug. So the pre-formulation studies keep on going simultaneously. After the complete studies have been done and no further fine tuning is possible, the formulation development is done, and then the as we discussed that right from the preclinical to the clinical trials it is done, and then large scale production and marketing is done. So formulating a prodrug is one of the part of these studies. Now, coming up to the first preclinical characterization studies, that is the physical characteristics. Now, physical characteristics are the characteristics which are related to the bulk properties of the drug, which are responsible for the for having the solubility as well as the stability of the drug. So, undoubtedly chemical properties also are responsible for the solubility and stability, but herein we are going to look at the physical characteristics of the drug from these point of view. Now, the first of these is, so in the bulk properties, the particle characteristics like shape, size, flow properties and bulk properties, that then porosity, density, hygroscopicity, crystalline polymorphism. Then in the solubility analysis, pK determination, solubilization, aqueous solubility, pH solubility, dependent solubility, partition coefficient, thermal effect, common ion effect and dissolution. Then what is the stability of the drug? So, solid state stability, solution state stability and drug excipient compatibility and stability analysis. Chemical characteristics, that is what are the various chemical reactions can happen with the drug because of the chemical structure, configuration of the drug. That is oxidation, photolysis, free radical reaction, hydrolysis, racemization, isomerization, enzyme decomposition and decarboxylation. Now, coming to the detailed bulk characterization studies, we would look into the bulk properties of the solids, like that is size, density, morphology. Now, this is important. This is the first step which is taken towards analysis so as to create a suitable and bioavailable dosage form. Now, crystallinity is the crystal habit and the internal structure of a drug that has an impact on the bulk and physicochemical properties of the drug, and this impacts the flowability, packing as well as chemical stability of the drug. Now, habit is the description of the outer appearance of a crystal, whereas internal structure is the molecular arrangement within the solid. So, how the molecules are arranged in the solid. Now, characterization of solid involves verifying that the solid is the expected chemical compound, characterizing the internal structure and describing the habit of the crystal. So, while we are going for the solid state characterization studies during bulk characterization, we need to first of all find out what is the crystal habit, what is the internal structure like, and whether it is the expected solid compound or not. So, as we can see here, there are different types of crystal habits like cubical, angular, spherical, granular. So, these are the habits. How does a material look like? The crystals, as I said, are the repetitive spacing of the constituent atoms or molecules in a three-dimensional array. However, amorphous moieties do not have this. So, the crystals are more thermodynamically stable in comparison to the amorphous forms. Amorphous forms are generally prepared by rapid precipitation, lyophilization and rapid cooling of liquid melts in comparison to the crystals which are slowly develop with controlled nucleation processes. Now, there is a possibility of transition of the phases of the habit of the drug. That is, there could be polymorphic transition, hydration, it may be it can from hydrated to dehydrated state and vice versa, solvated to desolvated state, then salt to the parent compound conversion or salt-salt conversion, that is chloride salt to bromide salt. So, salt-salt conversion is there, co-crystal to parent conversion or co-crystal exchange, amorphous crystallization to vitrification. So, all these thermodynamic stability or the destabilization of the molecules, there is a possibility of transition of the phases. Now, the crystalline compounds can have a stoichiometric or non-stoichiometric amount of crystallization solvent. Now, the non-stoichiometric adducts such as inclusions and clathrates involve entrapped solvent molecules within the crystal lattice. A stoichiometric adduct, commonly referred to as solvate, is a molecular complex that has incorporated the crystallizing solvent within the specific sites of the crystal lattice. So, compounds having water are called hydrates, whereas compounds not having water are called anhydrous. So, the crystalline compounds can have solvent molecules, then it is called hydrate. If water is not there, any other solvent is there, it is called as a hydrate. Now, these molecules, these water molecules or the solvent molecules could be included in the compound in a stoichiometric or a non-stoichiometric amount. So, if it is stoichiometric, you specifically know whether how many molecules of the hydrate or the solvate has been incorporated. If it is non-stoichiometric, it is called as an inclusion or a clathrate, and it is entrapped within the crystal lattice and it is not fixed. Polymorphism is a property of the drug to exist in more than one distinct crystalline species with different internal lattices. So, polymorphic form A, B, C, gamma, theta, delta. So, you might have come across these terms that same compound exists in more than one form. So, it is because of carbon, for example, right. So, it is because of the distinct internal lattice arrangement of the molecules. The compound exists in different polymorphic forms, and based on the internal lattice arrangement, its chemical and physical stability will also change. So, while we are going for a selection of the polymorph, while for the formulation development, we need to be careful that the polymorph which we are going to select is stable, has right amount of solubility, and it does not convert back it does not convert into a stable or a metastable form upon being subjected to a suitable temperature. For example, if on reaching a specific temperature point, polymorph A changes into polymorph B, and while processing you are using that temperature. So, there is high probability that polymorph A changes into polymorph B. So, that might be a negative point towards development of a stable dosage form, because you started with polymorph A, but you are getting a polymorph B which might have a lower solubility than the polymorph A. So, it is very important that the polymorphic forms of the drug are clearly understood, at what all temperatures they are stable, what is the solubility of each of them, what is the liability of their conversion from one form to another, so that the right form is selected. Similarly, with the crystalline as well as the amorphous forms, it is important to select the right form, because crystalline forms generally cause irritation to the mucosal lining. So, a very highly crystalline structure can lead to internal issues. Now, the polymorph generally exist in two forms, that is enantiomeric and monomeric form. Enantiomeric forms can reversibly change into another form by varying the temperature and pressure, and the monomeric forms, however, are unstable at all temperatures and pressure. So, we have to take care of which form we are going to select. So, the analytical methods which can be used for solid form characterization are microscopy, fusion methods, differential scanning, calorimetry, infrared spectroscopy, scanning electron microscopy, thermogravimetric analysis and resolution and solubility analysis. Now, microscopy is a technique using a microscope. So, we can use an optical microscope which is based on light, and based on that you can study the structure of the compound, whether it is depending upon what size you are taking, and resolution has to be changed. So, if this particles are very small, then we need to go to scanning electron microscope or transmission electron microscope. So, for cruder particles, even optical microscopy can be used for determining the structure. Hot stage microscopy can also be used, wherein the fluid phase transformation as a function of temperature is observed, and if there are transformations being observed, that is, the compound exists in polymorphic forms. X-ray diffraction. So, the diffraction pattern of the compound changes depending upon its internal as well as external structure. So, a globular moiety will have a different diffraction pattern in comparison to a angular or an oblong particle. So, the diffraction patterns will change depending upon the structure of the solid particle. Then within that there is the powder X-ray diffraction. So, the powder X-ray scan, even the powders, single crystal as well as powders can be used, can be subjected to X-ray diffraction, and their crystalline nature or the morphology can be determined. NMR. The NMR can also be used, wherein the powder sample is rotated at a specific angle with respect to the magnetic field, and the structure of the compound can be determined. FTIR, that is, it can also be used to determine the polymers based on the functional group changes. Dilatometry, microcalorimetry, thermal methods like DSC, DTA, TGA, melting point determination, all these things can be, all these techniques can be used for the determination of the original form, as well as if while change in temperature there is a change in form. For example, in differential scanning calorimetry, so that with change in temperature, what is the change in the melting characteristics of the drug can be observed. So, if the melting characteristics of the drug changes, that means the form of the drug is also getting changed. Now, clathrates are a single phase solid with two distinct components, the host and the guest. The guest is retained in the closed cavities provided by the crystalline structure of the host. Clathrates are generally determined using the melting techniques, wherein the because of two distinct moieties being present, their melting characteristics are different. Hence, you can determine whether it is a pure compound or it's a clathrate. So, with this we come to a partial conclusion of the pre-formulation studies, that is, these studies are basically required to create the basis for the formulation studies, and sound knowledge of the physical, chemical and biological properties helps in developing a suitable dosage form which is rationalized based on the physical, chemical and biological properties of the drug. So, if the drug is very poorly soluble and has poor permeability, you cannot give it orally. So, it is not going to going to be dissolved in the stomach, nor it is going to be absorbed. So, you have to have a dosage form which is directly injected into the blood. So, that is what it means rationally. So, the dosage form design is purely dependent on what are the characteristics of the drug and how well they have been determined. So, in the next few lectures we will be looking into other aspects of the physical, chemical and biological properties of the drug. Thank you very much for your patient hearing.